try and break it down a lot, and I've actually switched orders of some things and kind of omitted some things to try and make it as clear as possible. Also have a peanut challenge. Good. Going on. Everything's okay. Okay. So today introduces the idea of signal transduction. So, right, we already know that something external to the cell will connect with a receptor, right? And in the case of lymphocytes, it's an antigen connecting with the receptor, and then the receptor has to signal all the way to the nucleus for something to happen. So signal transduction is how that happens. And it happens in a chain reaction similar to when you study the complement system, in that a protein is activated, which activates another protein, which activates another protein, and eventually it gets to a transcription factor. So they mentioned some transcription factors like NFAT and kappa B, AP, STAT. I think were all mentioned, and these are just things that have the ability to basically turn on the DNA and start transcription for a new protein of some sort, whether it be a cytokine or anything. So in this case, though, in the case of complement, right, in the chain reaction, it was cleavage. Something cleaves something, which cleaves something, which cleaves something, which cleaves something. Here, it's all about phosphates. So it's about phosphates connecting with a protein to activate. And the things that do this are called protein kinases. So what they do is protein phosphorylate. They put a phosphate group on. And then when the signal is done, something called protein phosphatases dephosphorylate. They take that phosphate off. Okay? So it's a chain reaction that is propagated by these protein kinases. So kinase puts a phosphate group on, and then the phosphatases take it back off. Okay. Okay, so start with a picture on page 254, figure 7.1, which demonstrates two types of receptors in general. So one type of receptor has the extracellular part, right, that binds to its ligand, whatever it may be. And then it goes through the cell membrane, and then in the intracellular part, some receptors have this kinase right on the receptor, and that can actually directly propagate the signal. Other type of receptors, T and B cell receptors being two of them, are associated with other things that start the signal. So you have your receptor, and you have the same extracellular part that binds. You have the transmembrane portion, and then the intracellular part is actually just non-covalently associated or near the kinases that propagate the reaction, the downstream reaction. Okay. So they have a very nice table, figure 7.2 on page 242, that has all kinds of these examples of all these different proteins that can propagate the signal all the way to the nucleus. And we're going to talk about a bunch of these today. I don't think it's important to know exactly what domains in them, but hopefully you'll be able to place most of these by the end. Okay. So besides the proteins that actually propagate the signal, there's two types of proteins, scaffolds and adapters, that assist in this. So scaffolds are large proteins that can recruit all kinds of different proteins together, but in and of themselves lack any enzymatic activity. And then there's adapter proteins that are smaller than scaffolds that can link two proteins together, but in and of themselves lack enzymatic activity. So they have a nice picture here, figure 7.3, that shows a scaffold that gets phosphorylated and then brings all kinds of proteins into close approximation. so then they can enzymatically affect each other, but the scaffold in and of itself does not do that. Or here's GRAB2, which is an adapter protein, which is bringing sauce in conjunction with this receptor kinase. In and of itself, it doesn't have any enzymatic activity, but it's bringing these two in close approximation. Okay. So let's just jump to T-cell receptors and what's happening. Okay. So your T-cell receptor, remember we've talked about the alpha and beta chain, but the T-cell receptor with just the alpha beta chain is not enough to actually propagate a signal. So the T-cell receptor of itself recognizes on the outside, but then its close proximity to CD3 and zeta chains, and these are the things that actually propagate that signal, transduce that signal towards the nucleus. So CD3 is made of a gamma, delta, and 2 epsilons, and then there are 2 zeta chains. Now in some texts, depending on what you're reading, it includes the zeta chain as part of the CD3. So the T-cell receptor complex is the T-cell receptor CD3 plus zeta chains. Although if a text just says T-cell receptor plus CD3, well, it's including zeta chains in that CD3. Okay. And then on these CD3s and zeta chains are something called ITAMs. Now, ITAMs stand for immunoreceptor tyrosine-based activation motifs. And we're going to learn about something called ITIMs later, which have inhibitory motifs. But these ITAMs are what, where that initial phosphorylation takes place and where the initial signal propagation takes place. All right? All right. So in the case of T-cell receptors, what initially is going to happen, remember, so T-cell receptors have a co-receptor. It's either CD4 or CD8. And we know it's a co-receptor because this part of the receptor binds to the same thing the receptor is binding to. So in other words, the T-cell receptor is binding to MHC, and its co-receptor CD4 also binds to MHC. We've kind of talked about that before. And CD4 is associated with LIC. LIC is the first thing that kind of turns on, and it phosphorylates all these ITAMs on the CD3 and the zeta chain. Okay? And after LIC has phosphorylated, the ZAP70 comes up, and ZAP70 binds to the ITAMs that have been phosphorylated by LIC. So the first two steps are LIC, ZAP70. So LIC phosphorylates the ITAM. ZAP70 is zeta chain associated protein. So it binds to these ITAMs on the zeta chain. LIC, ZAP70. If there is anything to commit to memory, it's those first two steps. LIC, ZAP70. Okay. So once that ZAP70 is phosphorylated, its next move is to phosphorylate SLP76 and LAT, which are brought together by something called GADs, which in turn activates protein lipase C, PLC. Okay? So I'm purposely going very slow because this is very confusing, right? So LIC, ZAP70, and ZAP70 phosphorylates LAT and SLP76 held together by GADs, which then phosphorylates PLC. All right, so we're starting to get to some detail that you probably should start to remember, or at least have a good idea about what's happening here. Okay, so now once this happens, this is important because once that PLC gets activated, it cleaves something called PIP2 into IP3 and DAG. And once this happens, a whole bunch of things happen. So one of the things that happen is that IP3 increases the intracellular calcium concentration, and that calcium activates calcineurin, and then calcineurin activates the transcription factor NFAT. And remember, that's the whole goal, is to get that transcription factor. So once NFAT, that transcription factor, is activated, then you get gene transcription. Okay? At the same time, DAG, which is another part of HIP2 degradation, DAG recruits protein kinase C, which activates karma, and that ends up activating NF-kappa B. DAG also starts the RAS MAP kinase pathway, which leads to activation of AP1, a third transcription factor. All right. So phospholipase C, PLC, cleaves HIP2 into DAG and IP3. So it's that first really important step where you get now a giant cascade. So IP3 opens calcium channels on the endoplasmic reticulum that allow the calcium to enter the cytosol. So this is mediated by something called STIM1. Right. Okay, let's skip that part one. So here's one immunodeficiency that was pointed out today. So this is kind of a step away. But a gene called ORAI1, its products form the crack channels. And those are the channels that the calcium gets liberated through. A mutation in these lead to a form of skin. A mutation in this gene. All right. So once that calcium channel gets open, then it associates with something called calcineurin, which associates with something called calmodulin, which associates with NFAT, this transcription factor. And once that happens, this transcription factor, NFAT, can get into the nucleus and start its job. Okay? So calcium works through calcineurin, calmodulin to NFAT. Now this one I would definitely know because, right, there's two calcineurin inhibitors that we routinely use when we're trying to modulate the immune system, cyclosporin A and tacrolimus. Okay. That's kind of this pathway. IP3 increases calcium through a STIM1 mediated system through the crack channels, activates calcineurin, calmodulin, gets the transcription factor NFAT. Okay? Process 2 is DAG, which also comes from HIP2. Remember, HIP2 becomes IP3 and DAG. So DAG can activate RAS, which activates RAF, MEK, and ERK, the MAP kinase. Activated at ERK enters the nucleus and activates transcription factors, like I believe AP1 is also one. So AP1 is made of Phos and Jun. So the MAP kinase, ERK, can come in and it can bind Phos and Jun to make AP1, which is another transcription factor. All right. So RAS, MAP kinase pathway leads to AP1, which is made of Phos and Jun. second thing that happens. Okay, last one is that DAG can recruit protein kinase C, where it phosphorylates something called karma-1, which eventually, steps that I don't really think you need to know, leads to NF-kappa B activation. Now, this is a NEMO-dependent system. And remember, we know that NEMO, which we've studied before in TLR signaling, leads to a form of immune deficiency, NEMO deficiency. Okay, now remember, a naive T cell cannot be activated by just signaling through the T cell receptor, and this is kind of a safety factor, right? So not any cell can come up and present on MHC class 1, say, to a T cell and start the inflammation. So remember, it has also a co-stimulator. Now, a co-stimulator is different than a co-receptor, in that a co-stimulator has its own thing that it binds to. So the co-stimulator is something called CD28, and it binds to B7. A co-receptor binds to the same thing as the receptor does. So CD4's co-receptor binds to MHC. CD8 is a co-stimulator, and this co-stimulator is necessary in naive T cells to start the signal. Now, things like inflammation are going to upregulate B7 and CD28, so it's kind of another fail-safe to make sure the immune system doesn't go uncontrolled, unchecked. So there really should be inflammation for whatever's being presented here to activate a naive T cell, because then you're going to get this co-stimulator. Now this co-stimulator is going to act through something called PIP3, which is also going to allow phosphorylation of this PLC, which we've talked about, phospholipase C, which we talked about before. So it's going to strengthen this signal to allow this signal to go all the way to the nucleus. This is necessary in naive T cells. So this is just one more nice, what I thought was a nice diagram of the signaling pathway. This is on page 259, figure 721, right? So it's not only the T cell receptor, which is going to send signal through what two first steps?
Something and then zap.
Yep, that's 70, very good. Just got to say it over and over.
Yep.
And then CD28, right, is going to also activate phospholipase C, which is going to generate some IP2, IP3, and DAG. And the IP3 and DAG are going to, through the pathways we discussed, get these transcription factors going, NFAT, AP1, and NF-kappa B. Okay, do have it one more time. Okay. I'm just going to quickly mention that CD28 has a competitive inhibitor for B7 binding. So CD28 is a co-stimulator, but what binds to it more strongly is something called CTLA4. CTLA4 also binds to B7, and it's going to inhibit this whole pathway. And we want this, right? We don't want to live in a state of inflammation. We want this to be a stronger bind. So if there's inflammation, you have CD28 upregulation, you can get that co-stimulator and propagate that signal all the way to the nucleus. But as soon as that inflammation starts dying down, well, CTLA4 is going to be present, and maybe just kind of present when you don't have inflammation at all. And if something's presented, it's going to bind to B7 and send an inhibitory signal. Okay, not to be obnoxious, but I'm going to go through it one more time. Right, so CD4 Lick is going to phosphorylate the ITAMs on the CD3 and zeta chains. ZAP70 is going to bind to those. So Lick ZAP70. This is going to bind to that LAT FLIP76, which is going to bind to phospholipase C. Phospholipase C gets activated and cleaves PIP2 into DAG and IP3. IP3 increases calcium concentration, working through calcineurin, activates NFAT. DAG has two jobs, so it can recruit protein kinase C, which works through KARMA to activate NF-kappa B. It can also go through the RAS-MAP kinase pathway and activate a series of mediators which lead to AP1 activation. All right. Similarly, the B-cell receptor is a very limited intracellular portion. So the extracellular portion, of course, is very important. It recognizes the antigen. B-cell receptors are in close proximity to Ig-alpha and Ig-beta, which actually contain the ITAMs for signaling. So in this system, something called LIN phosphorylates the ITAM, and then SIC is the equivalent of ZAP70 here. So LIN phosphorylates the ITAM, and then SIC docks on the ITAM. LIN-SIC. I don't think there is a great next picture here. There's a better one in a box. But the same cascade is going to happen, but before, when PLC got activated by LAT, FLIP76, right? So the equivalent here is going to be blink in BTK. Bruton's tyrosine kinase. Do I have a separate slide for this? I don't think I do. Right, Bruton's tyrosine kinase, if there's a mutation here, well, then you don't have any B cells. So remember, a requirement for actually becoming a lymphocyte is you have to actually be able to signal through the receptor successfully for that lymphocyte to go out into the world. So if you have no Bruton's tyrosine kinase, you're never going to be able to signal successfully through this B-cell receptor. So you're going to have no B cells. The B cells actually can't be made because you're getting no correct signaling through. But this same complex is now going to do the same thing. It's going to lead to phospholipase C activation, which is going to cleave PIP2 to DAG and IP3. And then it's the same pathways. You get calcium liberation, NF-kappa B. You get the MAP kinases to AP1. IP calcium, NFAT. Sorry, calcium to NFAT. MAP kinases to AP1, and then DAG through protein kinase C leads to NF-kappa B activation. These are very similar, the same. You really don't need to know any differences here. Okay, now in the same way, you have a co-stimulator in T cell, you have one on B cell as well, and in this case it's CD21, which is also a complement 2 receptor, which binds to C3DG. So when the pathogen is getting recognized by the B-cell receptor, it probably has complement on it already, and that complement is going to bind to this complement receptor, CD21. And then through a complex of CD81, CD19, CD21, you're going to get activation of PI3, which again can activate that PLC pathway. So B cells, T cells. So T cells, LIC, ZAP70, and then those adapter proteins, LAT, SLP, all go protein lipase C. And B cells, remember it's the ITAM on Ig alpha, Ig beta, LIN-SIC, co-receptor is CD21, all to the same 3 transcription factors. All right. I'm going to kind of stop there for a second. Does that kind of make sense? Abbas has very good pictures for this. I think simplify it a little bit. But here you really got to see the big picture through all these little details. That I hope is a better big picture. Okay. So ITAMs are not unique to T cell and B cell receptors. You find them all over the place in the immune system. Lots of things signal through them. Here are a few examples. Okay. These are some antibody receptors, and now is a good time actually to talk about how these receptors are named. So antibody receptors start with FC because they bind the FC portion of the antibody. And then there is a Greek letter that signifies which type of antibody it binds to. So FC gamma receptors, the R stands for receptors, so FC gamma receptors bind to IgG. FC epsilon receptor binds to mast cells. Okay, FC epsilon R, and then there's just a designation 1, 2, 3 because there can be more than one receptor for each antibody. So in the case of IgG, there's 3, 16, 32, 64. 64 is FC gamma R1. FC gamma R2 is actually inhibitory, and then FC gamma R3, CD16. And then on mast cells, there's FC epsilon R1, the primary mast cell receptor. All signal through ITAMs. It's that same signaling process many times. Some, however, have an ITIM, which has an inhibitory motif instead of an activating motif. So if this receptor gets ligated, it's going to send inhibitory signals. So examples are the KERs, which we've talked about already a little bit in NK signaling. Remember, so when NK cells sense something, they're going to get a whole bunch of different signals, some inhibitory, some activating, and whichever one kind of and which one wins. So these are some inhibitory receptors, ITIM. Here's another inhibitory, FC gamma R2B, CD32. As an ITIM, it's inhibitory. So it's ITAMs and ITIMs to send these signals that are going to help cells decide whether they should act or stand still. So it's tempting when you're studying the immune system to think of everything as just a isolated event, but it's not true. It's lots of signals are occurring. In fact, what did they say? Maybe 50 different T cell receptors are getting activated at once to send this really powerful signal for something to happen. Just 2 are getting ligated. Well, you know, it's probably not going to happen. There's probably more inhibitory signals that are getting ligated at that point. Okay. Moving on to some cytokine receptors. So many of these cytokine receptors come as dimers. And when the dimers come into close approximation, then they actually can start a signal. So there are some that are homodimers, so the two chains are the same. Things like growth hormones and erythropoietin use this frequently. But then there are some that are heterodimers, and a great example of this is a cytokine receptor that one of them is a common gamma chain. So the common gamma chain is found on receptors for several different cytokines, of which I would probably memorize 2, 4, 7, 9, 15, and I actually think 21 is included, or at least Boss says 21 is included in that. Now these are very important growth factors for T cells and NK cells. IL-15 is essential for NK cell survival. 2 and 7 are essential for T cells. So if you have common gamma chain SCID, which we still believe is the most common form of SCID, it's X-linked. You're not going to have T cells or NK cells. So you're going to have T negative, B positive, NK negative SCID. IL-15, IL-2, and 7 important for T and NK cell survival. Okay, so when many cytokine receptors get activated, their cytokine binds and it dimerizes this receptor. So then it utilizes something called JAK, which does have kinase activity. They activate each other, they phosphorylate each other, and then something called STAT docks onto it. So it's a JAK-STAT pathway. Once STAT gets phosphorylated, then it can dimerize and it can actually serve as its own transcription factor. So you'll learn about different STATs like STAT3, STAT6, you'll probably read about. So it goes through this, what is seemingly very simple pathway of dimerization, JAK, STAT, and STAT can actually serve as a transcription factor and turn on certain genes. So cytokines, JAK, STAT pathway. All right. The last thing talked about in this chapter is apoptosis. So apoptosis is a form of programmed cell death. So what the cell is programmed to die, instead of just bursting open and causing inflammation by its death, what happens is the membrane reforms and forms blebs and takes little pieces of the cell that's broken down, packaged nicely to be processed by macrophages, so it doesn't actually cause inflammation as it's dying. Programmed and packaged in a nice and neat, orderly way to die. So this can be done by two pathways, the extrinsic pathway or the intrinsic or mitochondrial pathway. So the extrinsic pathway is mediated by something called FAS receptor, which binds to FAS ligand, SAS, FAS and FAS ligand, CD95 ligand. So FAS ligand will actually trimerize FAS, so there's 3 of them. And it clusters a domain, not an ITIM, not ITIM, ITIM, not ITAM. It's called a death domain. And this starts a caspase cascade. And I think this is one of the important things to remember about apoptosis. It goes through caspase signaling. So in this case, it recruits pro-caspases, and then it actually changes them to caspases, and then a caspase cascade leads to apoptosis. So FAS ligand signals through caspases to bleb and do this organized program cell death. Alternatively, if there is an insult to the cell, so they give examples, I think, of UV light or starvation. Then something called cytochrome C is released from the mitochondria. So this is the intrinsic or mitochondrial pathway. And when cytochrome C leaks out, it binds to APAS1, which then activates caspases 9 and 3, and then it goes through the same caspase cascade to start in a nice organized way, breaking up the cell into blebs, into small little blebs. So there's no inflammation in this process. So extrinsic is fast ligand, or CD95. Intrinsic, cytochrome C from the mitochondria, activates caspases, and then you bleb. So this mitochondrial pathway is mediated by this BCL2 family of proteins. So in my opinion, you don't really need to hold on to exactly what each of these things does. But when you see all these short V words, the BCL2 family of proteins, remember that they are part of the mitochondrial pathway of apoptosis. All right. So this is actually something I moved from the beginning. And we've talked about this a little bit before, but they bring it up again as this process of ubiquitination. I didn't like where it was stuck. I thought it made things confusing. I kind of dropped this down to the end. But this goes for receptors. This goes for almost anything that job is done in the cell that is ready to be discarded. So when that happens, something called ubiquitin binds to it, and this can happen several times. So you get polyubiquitination, and once that happens, you get this ubiquitin tail that signals to the proteasome to degrade it. So that's what I want you to know about ubiquitin. You can ubiquitinate something, and it basically marks it for degradation. And that's it. Any questions about any of that?
Oh, that helps a ton. I'm trying to. Yeah. The big picture.
See the forest through the trees.
Yes.
There's so many trees to look at.
Yeah.
See the big picture, especially in chapters like this.
And maybe we'll get to it in the next chapter. I don't know. But so what, like the transcription factors, you know, is the ultimate result, obviously. So what, like, are there specific
things it does?
Transcribing or
yeah.
That, or is it just?
So transcription, so there's lots of different transcription factors. And basically they just, you know, basically cling on to a promoter region of whatever. So in the case of T cells, typically going to be cytokine genes, right? So more IL-2. So it's going to turn on that part of DNA to start transcription through from that promoter through that gene. And remember, depending on the transcription factor, one gene can actually make several different proteins. So not even necessarily every part of that gene is going to be turned on. So it's a really sophisticated system. And this is kind of where it goes from molecular biology to genetics. But I think the big picture to remember is the transcription factor binds the promoter and turns on that gene. And think back to the cell of what you think it would be turning on. So right in the case of T cells, well, it's pretty much cytokines are going to get turned on in this situation. But not so for every cell. So some cells are going to differentiate based on transcription factors. So we'll talk about T cells turning into other types of T cells, like Th2 cells or Th1 cells. So they're going to turn on different transcription factors to to change other things, but, to make their own set of cytokines. You're going to change maybe the Tregs. You're going to turn on different transcription factors, right? We talk about STAT3 in that context. So you're just trying to get from the receptor, which we've talked about a little bit, through the cell to the DNA. And Beth's never really talked about genetics very much. I don't know if this one does or not. You should have like a Sarah Soden come in. She gives a great talk on transcription and translation. But all right, that's it. See you next week for Chapter 8. And then I think we'll get a little break because then we'll do a Jeopardy. I don't think we do that many more before the meeting, yeah, but they are pretty intense chapters right now.
Yeah.